மறுபடியும் ரெஃப்ரெஷ் பண்ணுங்க அது. மைக் மட்டும்.
Warm greetings to everybody in virtual and offline mode. According to the most classic Tamil language text, Tirukkural, dated 300 BC to 5th century, in the chapter Mei Unardal, Truth Consciousness, எப்பொருள் எத்தன்மதாயினும் அப்பொருள் மெய்ப்பொருள் காண்பது அறிவு. Meaning is, whatever thing of whatsoever kind it may be, considering only the true thing of it is the real knowledge. In accordance with this couplet of Tirukkural, Dr. Naim Hassan has rich knowledge in chemical ecology, currently working as Managing Director, Russell Biosolutions Limited, UK, which is a leading manufacturer and supplier of biorational pest control technologies to minimize the use of chemical pesticides and promote safe, sustainable, and eco-friendly pest management in agri and horticultural crops. Dr. Hassan has been involved with biorational crop protection for over 20 years and has specialized in insect pheromone, biopesticide, biostimulant development, field testing, and market promotion. He has carried out his PhD research work at the Natural Resources Institute of the University of Greenwich, UK. Dr. Naim has started his working with Russell IPM since September 2007 as a senior scientific officer. To date, he leads dynamic research and development team in the UK. As such, Dr. Naim Hassan is an accomplished entomologist with a wide range of experience in both the academic and commercial world in the UK and developing countries. Dr. Hassan has also been working as a visiting professor at Mediterranean Agronomic Institute of Bari, Italy for master's degree program. Dr. Hassan has extensive short-term overseas biological control research experience in Asia, Middle East, and Africa. Dr. Naim is a member of the International Society of Chemical Ecology, ISCE, International Organization for Biological Control, IOBC. He actively participated in many professional conferences organized by International Organization for Biological Control, IOBC, International Biological Control Manufacturers Association, IBMA, and other crop protection organizations. During his PhD studies, Dr. Hassan worked on the pheromone of the legume pod borer, a major pest of the legumes in African and Asian countries. He also won a fellowship to visit the International Center for Research in the Semi-Arid Tropics, ICRISAT, in India to carry out field trials of new pheromone lures and traps. I extend a warm welcome to Dr. Hassan to present his topic, Practical Use of Insect Pheromone for Climate Smart Crop Management. We welcome you, Dr. Hassan. Thank you.
Thank you, guys. Can you hear me nice and clear?
Yes, your voice is clear.
Thank you, guys. Thanks for a nice introduction. Welcome everyone to this session. My name is, as the presenter mentioned, my name is Naim Hassan. I'm working in Russell Biosolution. I will straight go to the presentation. I know that time limit. We have a 45 minutes presentation, then followed by the question answering session. If anything, if you have any questions, please feel free and ask in this question answering sessions. Let's share the presentation which I prepared for you guys. We go to my presentation. This is the presentation. Oh God, I don't know. Wait. stop share. Let me open it first on my desk there. So if I can go back to the Zoom again. Sorry, where we are. Can you see my screen?
Yes, sir. Yes, sir. It's visible. You can continue, please.
Can you see the presentation as well?
Yes, sir. Yes, yes.
Okay, that's great. Guys, today's talk will be insect pheromones practical use for a climate smart crop management. And before going to the main topic, I would like to give you a few insight about who we are and what we do in the UK. Rusol is a group of British companies working together for sustainable crop protection systems. We are one of the leading manufacturers of semi-chemical based monitoring and control products and at Rusol IPM. And Rusol Biosolutions also deal with biologicals, biopesticides, biostimulant, and biofertilizer. And also we have another company called Rusol IoT who works with digital innovations and smart technologies for smart monitoring of crop pest solutions. Company has been in the market over 30 years. We started our journey as an office spin company of Imperial College back in 1987. Then we gradually moved to our first office opened in Russell Square in London. Here's the name is Russell, come from there. We had a steady growth. Then we moved to the north of England and gradually to move to the north of Wales, the way the company headquartered in Deeside and at Flint. So we have been in the market over 30 years and we do market our product around 80 countries worldwide. And we have been awarded Queen's Award for Innovation twice, 2012 and 2018. And we have been awarded Queen's Award for Innovation, apart from innovation, international trade in 2011. So we have a young group of scientists and entrepreneurs together, and we are a company who invest heavily on R&D. We do research and we bring innovative products, then sell in the market and fund that research again. We really heavily, we do 50% of our profit goes back to our research again. And we possess some specialized knowledge. Therefore, we've been a part of various government-funded projects, Innovate UK, DFID-funded projects, and European projects. We have an in-house screening of pheromone. We can do the early screening of pheromone identifications, and we have some tools to do that, GCMS, GCFID, HPLC, and we have an insectary, and we can do the fundamental research at the beginning. So as I say to you, it is a commercial venture. We focus on customer need. We try to engage on customer problems rather than doing a blue sky research. And we start where the customers struggle. From there to upwards, from the problem to the development, not the development to the problem. We work our way up from the bottom up approach. These are some production facilities in the UK and the production lab for pheromones manufacturing. We manufacture over 160 species of pheromones and 10 to 15 different traps. And also we do also traps and pheromone impregnated with color traps. There is also biosolution. I'm going a bit fast. Biosolutions, as I said before, there's two sets of companies we have. One is dealing with IPM related to the pheromones, attractants, repellents, trappings, and pheromone related products. Our Rusol Biosolution deal with biologicals, like biological pesticides based on microbials, plant extracts, microbial extracts. Then we also have some attack and kill systems, which is putting pheromones and insecticide together. Biosolutions based in the Flint is a small town in North Wales, and we have a strong group here. I would like to show you this trend of biopesticide markets. It's a very interesting slide. And this trend is that, look, at the moment, growth of biological pesticides is 17 to 20% right now. But in this slide, you see by 2040, 44, the growth of biopesticide will exceed the growth or supersede the growth of chemical pesticides. So it's a good time coming ahead. So now today's talk about semiochemicals. Semiochemicals. What is semiochemicals? Semion is a Greek word, means signal. So these are the chemicals which plants and animals using to communicate with each other or their surroundings. They're very effective in its small quantities and they are environmentally friendly. So we can exploit these semiochemical based technologies for the crop and pest management. You come across pheromones, which most common semiochemicals, which mostly released by females, followed by male. And we have another groups of semiochemicals. It's the one first I showed you, that pheromones is we call it intra-species, same species, really one of the member of the species, either male population releasing female following or female releasing male following. But here is another group of semiochemicals, we call it chiromones, two different species. The odor of cow urine is giving message to the flies. There is a host there. So this is called chiromone, the interaction between two different species. Now, one of the use of the pheromone will come later on, called mating disruption or sexual confusion, which will come later on to bombard pheromone in the ecosystem and confuse the insect, though they are staying quite close proximity, but could not find each other. Now, where does pheromone lies in insect body? Pheromone released from the 8 or 9 abdominal segments through the cuticular hydrocarbons, and then it is perceived by the insect antenna. Antenna are very powerful. It can detect pheromone in very low quantities. Now, how do we get it out of the insects? We know the insect release it. So how do we isolate it? There's some of the techniques I would like to just giving a brief introduction. Like if you can put the pheromones, the insect in a container, and you continuously suck the air, which they are in their proximity. So this air containing pheromones, so like this containers full of midges. So whatever they're releasing, we put a suction pump and a filter. That filter absorbing all the pheromones. So these are the filters. We call it Poropac Q filter. So once the filter absorbs the molecule, you can wash it with the solvent, then inject on GC, and the gas chromatography will tell you how many molecules is there. So now in the practical aspect, we know the pheromones are the chemical signals organizing, really getting the releasing by or secreted by one individual of a population and detected by another individuals, giving you the specific actions. In here, for sex pheromone is for sexual orientation, what pheromone lead to. So chemically wise, chemistry wise, if we look at it, it is a complex mixture of straight chain acetate, aldehyde, and ketones. Normally 3 double bonds are called unsaturations. Most of the pheromones are like this, over 75%, but there are some odd one which has got 4 double bonds, and it's formed the carbon chain of C17 to C23. So use of pheromone in agriculture is many various use. We can use it for monitoring the population. We can use it for control. So monitoring and control. Control is the different kinds of control, which we'll talk later on. Mouse trapping, mating disruption, and attack and kill. So these are some standard structures. Z9 hexadecynyl acid, aldehyde, acetate. And when you look at the pheromone structure of an insect, there will be major component, there will be minor component. There is a ratio. This ratio is critical. If the ratio varies, insect will not respond to particular pheromone lures. So these are some lures, example of the lures. We used plastic to release pheromone, we use rubber, we use cellulosic filters, we use corks. It varies on the molecule and the insect pheromone ratios. So some of them are listed here, just give you some outlook, how they look like. Now we use rubber septa, vials, cellulosic filters, we use sealed tubes, wax emulsions, and even we use sponge or felt materials to release pheromones. So for monitoring, it's very important to monitor the pest population. So monitoring pheromone is used to monitor the population before putting a control action, or after applying an insecticide, how effective the insecticide application was. Monitoring will tell us. If we're capturing our trap, after applying a pesticide, it means the pesticide wasn't effective. And also monitoring can be used for determining threshold of a cropping system or controlling a pest. And it's very important on quarantine pests. Those pests are not present in the country. If you have a detection traps, that will tell us whether the movement of that particular pest. So monitoring is important to know what's going on in your field, to determine the action, and after action, how effective the action was, and to map out the population movement. This is a simple trap we can use for monitoring, a delta trap with a sticky surface and a pheromone. One of the important thing is here that when you use a delta trap and a sticky surface, you can visually see the insects. The insect is visual. And if we use a water trap in this monitoring, then it will be difficult to physically see the insect, an entomologist or taxonomist to identify which insect it is. So always we suggest use a clean delta trap or a sticky traps for monitoring. There are various types of traps. I'm giving here an example. There are various pheromone traps based on the physiology and behavior of the insects. So for moth, we use bucket traps, moth catcher traps. For flies, we use fly catcher traps and also delta traps. These traps is a new innovation from us. You call it ferrolite traps, where pheromone and light together. Pheromone is attracting male population of an insect, and the light is attracting female. So by using light and pheromone, we can achieve better control. There are some pictures and some images of the pheromone being using in the field. So for example, when it's a dusty condition, we do not recommend to use the sticky surface of a delta traps, and we rather recommend to use a moth catcher traps. When it's a clean climate, we use delta traps to monitor the pest population. Now I'm going to talk away from the monitoring, a control system about mating disruption, which you are familiar with. So mating disruption is a system where we disrupt the mating. So one example is European grapevine moth, Lobesia butrana, is a severe problem in Europe. If there is no control measure is untreated, lead to 40% crop loss. And traditionally growers spray chemicals, but they are not working well, and there are residue problems. So mating disruption been explode in Europe last 30, 40 years, and is a very robust system where the grapevine moth become completely confused, and they stays close proximity, couldn't find each other. And this system in a valley in Italy, there's one valley, Trentino Valley, in the north of Italy, 60,000 hectares of grapes. So this area is mostly covered by mating disruption. Then apple areas. But one good thing we need to bear in mind, mating disruption only works where it's a monoculture, a complete culture of same crop and same pest. And the minimum requirement of adopting mating disruption is about, I would say 5 to 10 hectares. If it is less than 10 hectares or 5 hectares, the system will not work. There will be intervention from outsider, immigrants, insect from the next door. So bear in mind, the mating disruption has to be a monoculture, same crop, a particular area. And this sector is growing. And there is some example of mating disruption dispensers. We have seen the monitoring lures, and this is slightly different. These dispensers are like a capsules, like a PB ropes, like this is a puffer, and this is a sprayables. So you can use various dispensers. Here, the main idea is to release a high volume of pheromone. There's 3 ways we can confuse the insects. When the high abundant amount of pheromone blowing in the insect antenna, insect become completely confused. And you know, the mostly moths, they don't eat a lot. They have some nectar or some drinks. So they bring the energy from their pupal stage. So this moths frantically moves, and they have a false tail. They could not find the natural call of a female. But this male is moving around and could not find her. So eventually they get completely exhausted and they die. And other thing is, when we blow a higher amount, it desensitizes their antennal action. The antenna become completely saturated, so it cannot even respond to their own pheromone. And other thing is, action, there's this, the artificial pheromone mask the natural one. Okay? Give an example. If you use the body spray or scent, but if you use 4 or 5 scent on your cloth, you will be confused which one is which. Similar ways, when we put higher amount, insect get confused. So this technique works. As I said before, it has a minimum area. So next practical use of the pheromone for control is attract and kill, or lure and kill. In this control system, we bring insect towards a point or a bait, near a bait station, where they come, touch the points and got the lethal dose, either die immediately or fly away and die later on. Give an example of here. You see, this is a fly, just as a metaphorically fly coming. This is a bait station which contains female attractant, hydrolyzed protein, and alpha cypermethrin. So they get excited, touch it, dead. Because alpha cypermethrin is instantly kill them. So we have developed a attract and kill systems for male and female Mediterranean fruit fly. The male is responding to the trimaglure and female is hydrolyzed protein. In both cases, you see here is a bait station, which is a sponge or a felt, which absorbing hydrolyzed protein and alpha cypermethrin. And here is a wax emulsion. Wax emulsion is containing trimaglure and abamectin. So one is targeting male, this one, and this targeting female. How it works, is a short video, have a look. These flies is an example how system works, are attracted to methyl-eugenol. So this wax emulsion here containing methyl-eugenol and abamectin. So flies thinking is a odor they like, parapheromone, so they're going towards it. And those of them already touched it, got the lethal dose and dead. And those who haven't died, they are hovering around. And you see these flies already dead. So farmer will see the kill instantly there. So it's not capturing in a trap. And in this system, we don't need to use a trap, just a point source. And these are the dead insects. So we move on. Next one, similar system. The way you've seen in the leaf, I put a wax emulsion here. The wax emulsion we put on a plastic dispenser, like a blister pack. Here, the reason of putting on a dispensing pack is like, rather than putting on leaf or bark, because it contains pesticide, we would like to say that pesticide is not going to the leaf or stems or bark. So it's here, and also chemically it's perform better. So if you look at this video, like small video, you see here, flies are getting in. It look like they are hovering around. Look like hives. So many is getting there. It is, and either they're touching it or they die there or they die later on. So let's move on to the next slide. So these are the techniques we used, male and female attract and kill. With that also we found that the fruit fly pupate in the soil. So we use this entomopathogenic fungus to kill the soil, the people population in the soil. So these are the some of the trials we have conducted in bitter gourd and also in mango by using male and female. How do we apply male and female? First, we apply all the bait stations for female across the orchard. And then we apply the male around the border. So the idea of protecting the border, those who are entering from outside, they get killed. And also it drives all the male from the center of the orchard towards the border. So it deprives females from the male, and females also going to the bait station. So both ways we controlling male and female. We have conducted a trial in guava in Bangladesh, and we found over 88% control than untreated plots. And we found in bitter gourd over 91% control. Both, and is a control without spraying a single droplet of pesticides. So these are another trial we have done in cucurbit fruit fly. We got 95% control, significant level of control. As I say to you, the entomopathogenic fungus Metrorhizium anisopli can easily grow on insect population. And we have conducted a lab trial to see how much it kills larvae and pupae, different dose, and we found over 60% larval and pupal population mortality in the lab. Then we have conducted the trials on adults, and we found 50 to 60% mortality on adult population. So this is the technique, that one I've showed you before, the attracting towards the point source, getting and killed. Now, that's called attract and kill. There's a new techniques we bring in, it's called attract and infect. Rather than using a pesticide, here our idea is to transmit. We know the entomopathogenic fungus can kill the larvae and pupae and adults. So if we can disseminate the entomopathogenic fungus, then it will reduce the population substantially. In this way, we developed a product called auto dissemination attack and infect system. So this is the fungus, which is the formulations entomopathogenic fungus we developed, and then this is a lures, and this is a bait station where this contain the spores, and this here we hang the lure. So lure is bringing the flies there, and they are getting the spores. In this here, they are not immediately get killed. They're collecting the spores, then they fly away and have mating and have mate with their mates, and they're also distributing to others. So this way we maintain a horizontal distribution of the fungal spores. There's a trial we have conducted in the UK on strawberries in polytunnels. The strawberries here, the SWD, Spotted Drosophila, is a serious pest. So we're trying to control this. You see this side, right hand side, there are spores, and when the flies get in, they get the spores and they'll get out. So we are not killing them here. We are giving them opening to go away and distribute the spores. So they are naturally spreading the spores. Eventually, the spores will germinate and kill them and kill their mates. So another important aspects of us to, if you look at the insect behavior and biology, around 60% insect pupate in the soil, like thrips, like Tuta absoluta, like Pholomium, like fruit flies. So in this aspect, this is a very important information for us. If we can tackle the problem when it's in the soil, that will be a lot more easier than when the problem bursts in the later stage of the crop's life. So therefore, it's important to target soil population. So if we can increase the number of Metrorhizium or Beauveria bassiana or any entomopathogenic fungus, then we will have an upper hand on insect control. So therefore, the attract and infect gives us a new clue that you can use chemical pesticide for attack and kill, or you can use biologicals, microbials, and spread them around. So they will spread themselves. And also if we drench or if we apply soil treatment, that will also reduce the population early stage, and you will not see the surge of a insect going later crop stage. So now I'm going to talk about another new techniques of using pheromone. It's called push-pull. It's a novel system where we will use push and pull to control harmful insects. So how it works, like giving you a scenario, the Ligas rugilopenis is the biggest pest. Is a capsid, is a problem in strawberries, raspberries in the UK. So we use a repellent to repel them, push them towards the border. And we use a pheromone, a trap or capturing system around the border. So the pull is by the pheromone and push is by the repellent. So in this climate, we conducted some field trials for 2 insects. One is for thrips, from Western flower thrips, which my colleague Claire has spoken to you. And now I want to show you how the push-pull works here. Thrips is attract to the color, blue color, and also thrips Frankinella occidentalis got a pheromone. So we impregnate pheromone or infuse the pheromone into the sticky surface. So all these blue rolls with the pheromone capturing, pulling thrips, and inside we use this is called a product called Magipal. Magipal is a repelling the thrips from the main rows towards the border. So this way we can see the substantial reduction of fruit damage and number of thrips per flower. If you look at this graph, there is a push-pull is a number of thrips bronzing, and capsid capturing is significantly different than the control treatments. And if you look at the mean number of thrips per flower, this minimum in push-pull is reduced over 90%, and is a significant difference. And similar we found in when we worked in raspberry. These are blueberry midges. Blueberry midges we use push-pull techniques. So when you use control, and if you see the push-pull, the population is always low in push-pull, and it is significant difference. So here, rather than using one single tool, we are using two tools to drive them away, pushing them away, then pulling them from the order. So these are the, so far we have done, developed strategies for various insects, open field and greenhouses, strawberries on thrips, capsids, blueberry midges, pear sucker, whiteflies, cabbage whitefly, tomato leaf miner, and also the date palm weevil. So date palm weevil, I would like to show you a few slides on that. Date palm weevil is a serious pest of dates. Is also a pest in India on coconut and also date palms. So we tried push-pull in date palm weevil, and we found that the techniques works, and it gives a significant difference when we use traps around the border, and we use this Magipal, and we use two rates, half a dose, double dose, and single dose, and we found a significant difference capture on control and the treated plots where the repellent is used. So repellent here, if I give me back, pushing the trap capture is very high in these traps compared to this. Okay, so what is happening here, the repelling effect of Magipal is pushing it, increasing the number of trap catches higher in the border. If we take this away, then you will not have the capture like this. So this is another innovative idea of bringing two systems together. So these are the captures. And now I am nearly done. I'm coming to the right up to the end, that how these systems works. So this system works, in conclusion, pheromone-based control strategies are why I said climate smart, because here we're reducing insecticide substantially. We are not doing any blanket spray. We are not spraying over foliage. We are giving a point source where insect will come, try and get the lethal dose and fly away and die. So pesticide is 95% less. So all the manufacturing of pesticides, chemicals, the logistics, the solvents, the factory running, the carbon emissions all get reduced. And also we cannot rely on the synthetic chemicals day in, day out, because after 4 or 5 years, we see the resistance building. So this is not sustainable. But in these techniques where we're using, we are talking as sustainable systems, where we're using natural clues and using it for the control of insect pests and diseases. So it can be used on its own, or it can be used with a IPM program. So pheromone is very compatible with biologicals, and some places it can be compatible with chemical insecticide. For example, we are doing a trial on rice yellow stem borer in Bangladesh and also in Nepal, and we are doing rice stem. There is a different stem borer, Chylosupra in Spain. So if you want to control rice yellow stem borer, the farmer's going for 10 to 15 spray. But if there is a mating disruption in place, they might need 1 or 2 spray. and they might not need sprays. So in that angle, it is a good fit with biologicals and chemicals. At the very end, would like to say that it also cutting pesticide entry to our food chain. As you know very well, in India, people spraying chemicals, the next day they bring into the market. There's no waiting time for our farmers. Farmers are busy people. It's their livelihood. So what they do, they go to the shop, guys, what do you have? I have this bottle. Go and take and spray it. Your pest will go away. And they're not reading waiting time. They're not reading the residue. So they just spray it and they control, and they got rid of the pest and they're selling it to us. And this way, heavy metals, heavy molecules is penetrating to our body. So therefore, the surge of high blood pressure, stroke, the cancer is very high due to the pesticide toxicity. So these techniques will cut down that, and it will work for us, will work for the consumers, and work for the farmers himself, because farmers get the fast lethal dose of that toxicity because he's spraying. So in this angle, by putting everything together, we can really build up a sustainable, climate smart crop protection systems. By this, I am finishing. Thank you very much for your attention. If you have any question, please feel free and ask me.
So, thank you.
I just seen a question on chat box. So can push-pull be used in all field crops and control? Yes, most of the crop is possible. And we have to look at what we're going to use for push-pull. If there is a pheromone identified, yes, it is. If there is no pheromone identified, it might be we will look into a chiromone or a kind of an attractant. Then it can be used. But if you look at, there are, for example, if there is no, it is usable in most of the insects, but some insects pheromone may not be identified. There will be a struggle. On that angle, you can use color traps, or you can use some other systems to capture it. Anything else?
So, thank you, Dr. Naim. And the participants will be asking the questions in the chat box. We will be waiting for some more time.
Yeah.
So, is it okay for you to view the chat box and respond?
Yeah, I can view the chat box. I can see that some of the comments and some of the questions.
Yeah. Because or else I'll coordinate. I'll read the questions to you and you will be answering.
Yeah.
That will be good, or you will be seeing and answering. Which way will be convenient?
Both ways. If I miss something, you remind me.
Yeah. Okay. Thank you.
No problem. Okay, I've just a question here about, please suggest one of the IPM model for Western flower thrips in chrysanthemum to keeping below economic threshold level. Okay. Western flower thrips is a major problem. We know it is a problem, and Western flower thrips can cause you severe damage. In chrysanthemum, we have an experience of chrysanthemum grows quite a lot into the flower business in Kenya. They export in Europe, so they cannot use willy-nilly any chemicals. So what they use, you use the push-pull like a pheromone impregnated sticky boards or rolls in greenhouses, glass houses, even open field they use it. And also you can use as IPM, you can use Metrorhizium anisopli or Beauveria bassiana in the soil. It's very important. And because then the soil population will be dropping down significantly. Then later stage you will not have adults. And also if you use these two things, the trap with pheromone will control the adults, and the pupae will be controlled by the Metrorhizium anisopli, then you will have good level of control. On top of it, if you want to release the natural enemies, that will also help to get the population down. But on our trial, we have seen if you use push-pull, like a Magipal and traps and soil treatment, you get over 90–95% control. There's another question is this possible to using push-pull techniques in storage pest management? Yes, it is possible. Storage pest management, mainly in storage, you have pests like tribolium, capra beetle, biscuit beetles, these beetles, and you have the moths like those on the, like an Indian meal moths, Plodia ephistia, these moths. So if we take that example of beetles, beetles, you can use before putting the grain, you can use some sort of bait station near the wall or near the peripheries where it will have pheromone and microbials. Okay, like a sort of so they will come and they will take it and bring it to their heap of grains, and then when they mate, they will give each other. And also in the UK, what we do before we bring in the grain in a silo or in a storage, we apply matresium. So matresium built up before grain coming in. So if any overwintering population in the storage, they come in out, they will get the matresium, they will die, and they will go to mortality. But if after putting grain, if insect, if you have the, as I said, you have the pheromone impregnated small bait station or balls around the border, then it will encourage beetles to leave the crop and go to the pheromone source. So this way you can manage it. One of the question is available to the field. Okay. Okay. To then improve the natural enemies. Yes, that's a very interesting question. Is there any pheromone available to improve the natural enemies population under field condition? Great question. I love it. What it is, the natural enemies, if we see the natural enemies behavior in the field, the natural enemies is always stick to the border, and they from the margin to the center, they move slowly than the pest moving in. When the pest population is higher and natural enemy is not cope with them, then the control fails. So if we can bring the natural enemies number higher than before pest coming in, then we have our guard is ready to catch the thief. So we have in Russell, we have developed a natural enemy attractant, and that attractant is like nothing but a volatile component. When insect attack the plant cry out for help, and in that plant release some volatile molecule like salicylic acid, abscisic acid, and jasmonic acids. So we developed a dispenser, which is natural enemy attractant, in this before attack. Like for example, insect attacking the host, then host releasing these signaling molecules to give the natural enemy message that I'm under attack. You better come and help me. So what we do, we take that volatiles before pest invasion. So these volatiles in a dispenser, and we put in the field, and giving message to the natural enemy, congregate more. So this way we increase the number of natural enemies, and with a higher number of natural enemies, there is a good chance of controlling pest without using pesticides. Can you manage plant parasitic nematodes organic way because of commercial nematodes high price? Yes, of course. Plant parasitic nematodes, nematicide is a major demand. We see there is a nematode problem in many crops. So there are funguses and bacteria which works very well against nematodes, like Bacillus firmis and also Paecilomyces linaceus. These two funguses are working very well with the root knot nematodes, especially melaleucine and other nematodes. So we have conducted a trial of banana root nematodes, and we found a significant difference with using Paecilomyces linaceus. So organically, both are entomopathogenic fungus and bacteria, and you can use them. And these products are, I think, available maybe in India and some part of the world, where you can use this entomopathogenic entomopathogenic fungus as nematode pathogenic funguses, and combination of bacteria and fungus. Other thing is good root health is important. If root is good, then attack of nematode needs a wound to get in. So if you can maintain good biostimulant and make the root good health, good health, and also use this bionematicide, then we can control nematodes organically. As a person, what will be the impact of the biodiversity if there is a continuous use of pheromone, as it will deplete the source of natural enemies? Well, that's an interesting question. Pheromone we are using inside the crop field. We're not disturbing insect in wild habitat. Okay, so here we're using pheromones to confuse them or avoid the mating. So we found that if you're not using wild habitat, so that it will not affect the biodiversity. And if even natural enemies have got different hosts, they're not relying on one single host. So we are protecting them. And so far, pheromone been used in so far commercially 50, 60 years, and we never seen any effect on biodiversity. Those are natural enemies. They are there, and in the wild host or non-crop plants, so it is there. What kind of solvent do you use in chips in pheromones? Okay, when there's a two different scenario. When you're using pheromone lures, we use solvent to dissolve a small amount of pheromone and to dispense this large area. But when you're using sticky traps, we don't use solvent. We use a emulsion. We make an emulsion. Then that emulsion goes to the sticky surface. And these are the techniques of our know-how, and we know how two polymers works together, because sticky glue is a polymer. And another important thing is mixing two polymers together and seeping through the glue is not easy. And also glue, when we glue the sticky surface, we coat it, the rolls, the glue is heated. So we have to find an area where heat is reduced, then we apply the pheromone. So it is a techniques where you need some professional help to get the glue and pheromone together. Can you use pheromone in field condition during rainy season? Yes, pheromone can be used during rainy season. Rain does not affect pheromone, and most of the attack and kill system or mating disruption are rain fast. Because the pheromone, bear in mind, pheromone is one thing I should have touched it, but I because time constraint I didn't touch it. The release rate. The release of pheromones depends on the temperature and moisture. Temperature is a major drive. So temperature in the rainy season, if the temperature is low and the pheromone release will be slow. If it's a very high temperature, then pheromones will be faster. So average pheromone last in the field dispensers are 4 to 6 weeks. But if you use wax emulsion, or if you use some sort of polymer, a slow release material, then it will remain effective for 3 months, 4 months. And rain does not affect it. It does not wash away, or it not gets because if you use dispensers, pheromone is inside the dispenser. If you use a wax emulsion or you use some sort of polymer, it is impregnated inside. So rain should not use it, dissolve it at all. Per hectare recommendation for such as fertilizer, tomato pinworms, thrips. That per hectare recommendation is using it depends on the strength of the product. If the spore is 1 times 10 to the power 7, then per hectare recommendation is 1 to 2 kg per hectare. And if the spore spore count is low, then we will say you have to use more product, more like a large, like a higher dose. Major issue in India, what common white grub? If I know the scientific name, that will be easier to comment. But white grub metrorrhizum works very well. Fungi works only confined environment like greenhouse, or no? Auto insemination fungi works in open field and greenhouses both, because we have conducted a trial in fruit fly. We have seen that it is the reason I've showed you the example of greenhouses, because in the UK we have the spotted wing Drosophila problem in greenhouses, but in developing countries, when in Kenya we have problems in open field. So you can horizontally, even scientific literature you can search. There is a ECP, a group of ECP, they did some research, and they found it the horizontal dissemination is possible in fruit flies by using Metrorrhizum anisopri. The one question is, salicylic acid and jasminic acid are deterrent substances developed in plant in response to the insect damage. Those stop insect feeding. How can they? Salicylic acid is a good attractant of natural enemies. You know, deterrent means deterrent for pest, deterrent for harmful insects. There is a nice combination here. The salicylic acids or jasminic, like we call plant active volatiles. When plant is under attack, even when the fungal disease happens, it also releases some volatiles. So these volatiles giving message is deterring the harmful insects, but at the same time, it attracting beneficial insects. And you look at the literature, you'll find in the literature is quite a lot of literature there that salicylic acid is an attractant of the beneficials. And we have seen it. We have when we applied recently, one of my PhD students, she worked on white flies and also on thrips in greenhouse conditions on peppers. And what they found, when we put them, when plant is starting producing salicylic acid, and we release insects, white fly prefer non-treated plant rather than treated plants. They doesn't want to go there, and it's a big difference, about 70% less suppression. And then we released natural enemies, and natural enemies prefer the one has got triggered release of salicylic acid. So it is a proven technology. I'm really sorry, I don't know about there is white grub pheromone yet. I need to check it whether it is identified. If it's not identified, it can be identified. Okay, and this Holotrichia serrata. So we can be done, but and so far my knowledge, I don't know whether any pheromone has. I need to look at the literature. But if it's been identified, we can work on the formulations. Our main strength is the formulation. If it is not identified, we can identify this pheromone, but it's need a long study of 2-3 years to identify the pheromones. Any other questions I missed, Silva?
So I hope you answered all. It's not there. Nothing is left. Nothing. Yeah, it's nothing left. I'm just looking for the cousins. So I hope there is nothing left. So it's a great session, Dr. Naeem, for having you with us. Because in the 11th hour, we have conveyed the schedule of the meeting to you, and you readily accepted. And actually, I conveyed the progress to Dr. Naeem in the evening. By around 8 p.m. in the IST, he just mailed me that he is principally okay for presenting the talk. So, and he conveyed the topic within another day. And it was nice to have you with us because the audience is majority graduate and undergraduate students. So it is a great motivational thing for our graduate students. So
Thank
that's
you.
why I was so particular about taking you along with us in this IQA 21. And you have been, we are fortunate to have you with us this afternoon.
Thank you. Thank you very much. If I know the students, next time, if we have opportunity, we can talk about how to isolate it, how to identify it, how to dispense it, how to physically see the effect of pheromones in the lab on insects, in wind tunnel, in white tube, in electrophysiologically. I can talk a little bit about that, which we teach in our MSc students in Bari. So in maybe next time, if we have an opportunity like this, we can talk about basics that which will help students to work out and develop their own formulation in future.
Thank you, Dr. Naeem. I take this opportunity to thank Dr. Shams Usmani also for coordinating things, because he has coordinated nicely and he made things to run smooth. Thank you for both Dr. Naeem and Dr. Usmani. And already as told by Dr. Nalini in the introduction that Dr. Naeem is a product of NRI, Natural Resources Institute, which is supposed to be a pioneering institute in bioproduct research. So we do have a very strong research setup here in Annamalai for biocontrol and as well as botanical insecticides. So earlier when I am talking with you, I have told you that my research is also from phytoinsecticides. We do have our Professor Dr. S. Arudin MB, Professor and Head Department of Entomology, who is actually characterized a molecule, and he has been doing consistent research on phytoinsecticide for the past 35 years. So it is nice to have you here. In the, your physical presence here will help us a lot, and I hope that we can materialize that by next year. I hope so. Yeah.
We will benefit from you as well, from your research, and we will work together to get benefit from there as well.
Thank you. And,
thank you.
Once again, I request Dr. Nalini to propose formal vote of thanks.
Dr. Hassan, on behalf of IQA 21 team, I wholeheartedly place our sincere thanks for your elaborate and lucid lecture on briefing about Russell IPM, biosolutions, biopesticides, semiochemicals, in specific isolation of pheromones, monitoring trap, pheromone mating disruption, and also interesting lively videos on Medifly, Batocera dorsalis, auto dissemination of metarhizium fungi for fruit flies, thrips, push-pull novel pest management with attractive illustrations. Thank you once again for your painstaking efforts to give us a visual treat and enlightening us about current advanced sustainable technologies in pest management. Thank you. Thank you.
Thank you. It's a pleasure. Thank you. So can I leave now?
So Dr. Naeem, actually, your session is over.
Thank you.
But if your time permits, you can.
No. I got to go. Thank you so much.
Okay. Thank you, Dr. Naeem. Bye. So actually the next session and the next speaker is Dr. R.S. Suresh Kumar, National Manager Product Development from Razi Seeds. We have some, because it is a hybrid mode presentation, Dr. Suresh Kumar will present his lecture here on this venue, and it will be broadcasted live as I have told you in the morning. So we need some 5 minutes break for arranging things. So I request the participants online to remain and participate in the discussion. So just a 5 minutes break so that we will be again into business.
Warm good evening to all. According to the Kural, Urudundu Varvare Varvar Matrellam Toludundu Pin Selbavar. It means they alone lives whom live by agriculture, all others lead the dependent life on them. Dr. Suresh R.S. Suresh Kumar hails from a traditional agricultural family with generations of practical experience in farming and farm produce management. Dr. Suresh graduated from Mahatma Phule Agricultural University, Maharashtra, and obtained his master's and doctoral degree in agriculture at Annamalai University. After this, he built his career in both academia and industry. His academic experience includes a short stint at the prestigious TNAU, Coimbatore. His professional experience includes over 24 years of progressive career growth with international market leaders such as Syngenta, Monsanto, DuPont, Pioneer, and currently heading product development function as national manager in Rasi Seeds Private Limited, Coimbatore. Dr. Suresh has been an integral part of research, development, and commercial initiatives in these companies. Throughout his career, he has built up his reputation as an outstanding and well-organized technical savvy. Dr. Suresh held a range of research, development, and training activities. He has built up a wide network of contacts and key opinion leaders in the field within academia, regulatory agencies, agricultural industry, and farming community. He has multilingual proficiency in English, Hindi, Tamil, and Telugu, extensively travels across the country and well-versed with cropping pattern of our country. He has attended various workshops and training programs in national and international institutes on agricultural technology. He has thorough knowledge in creating communication tools on crop management. I extend a warm welcome to Dr. Suresh Kumar to present his topic, Insights on Insect Resistance Breeding. I welcome you, sir.
Thank you, Dr. Nadu. Good evening and greetings to organizers of this international workshop, respected professors, graduates sitting at this auditorium, and greetings to the online participants. And I'll be taking you through insect resistant breeding for the next 45 minutes. And my co-presenter, Dr. Kannan, principal breeder of Rasi Seeds, was about to join, but unfortunately, due to the flight schedule, he is not in a position to join. Our sincere apologies because he is the expert who can handle this presentation in a better way. Still, I'll try to do justice in making this presentation in a better way. And going to the topic, before starting insights on insect resistant breeding, I want to touch upon the host plant resistance, which is the core and foundation of insect resistant breeding. And when I see this slide, this host plant resistant, always my memories get flooded with my PG HPR course, and fortunately my course teacher Dr. Selvanarayanan is here, and painter's definition, the relative amount of heritable qualities that influence ultimate degree of damage done by insects. Still, I remember that painter, that was the first definition of first class of postgraduate course of host plant resistance. So this picture usually we use for representing and illustrating the heritable qualities. Usually They use double-stranded DNA helix structure, but we love to use this picture because of the pollen flow of Gerbera. It is also a kind of heritable qualities, transfer of heritable qualities. So this we usually love to have this picture. And moving to the insect resistance breeding, the plant defense mechanism. Usually the broad 5 topics of insect resistant breeding or insect tolerance breeding start with target related insect studies. Even I can share the very recent memories of our PhD. I had field reaction studies against a major insect pest of maize, and in that 5 years of study, myself and my chairman, Dr. Nambi, we spent almost 2 to 3 years in collecting and collating data only on the insect part, the hotspot area, the insect population dynamic, pest population dynamics, and the life cycle and the influence of other parameters. Then after thoroughly studying that, the insect related studies only, we moved to study the field reaction of various cultivar, popularly cultivated species of corn in southern India. So that is the basic step and basic foundation. The first step is the target insect related studies, and second is the carrying out the insect resistant screening in various germplasm. Various germplasm means the cultivated germplasm or the wild species. So various germplasm, usually this is the breeders' tireless effort, where they used to screen this germplasm through selection and from the germplasm pool on this insect resistant screening. Then after screening a suitable germplasm, they identify the mechanism of resistance in the selected germplasm. That is the third step. And the fourth step would be the identifying which gene is responsible for the gene or trait of interest. That is again the breeder's interest, the fourth step. And finally, the final product outcome is the resistant or tolerant cultivar variety or germplasm will emerge out after 7 to 8, minimum of 7 to 8 years of tremendous work of breeder. Then it will be screened across the various agroclimatic zones to validate the field reaction of these germplasm against the target pest. And the due course, after introduction of the variety or hybrid or a cultivar, again after 5 years, again a gene pyramiding or improvisation will happen on this germplasm. So the broad classification of plant defense mechanism, the insights of insect breeding is the target insect studies, insect screening in the germplasm, identifying the resistant mechanism in the germplasm, then identifying the gene responsible for the insect resistance, and finally giving a product and screening the product and monitoring the efficacy of the product for the longer life cycle. So I'll be quickly taking up through these 5 topics in my presentation. And of course, after, so I'm not touching more on the insect related studies. So I'll be covering the next topic, because we all entomologists, we know on the pest related, pest population dynamics, the life cycle studies and the weather parameters and all. So I'll be skipping to the next level on the identifying the germplasm. So the source of the insect resistant germplasms are wild species, and the cultivated species, usually the varieties or hybrids we usually cultivate in our agroclimatic zone. And the third is germplasm pool in the public or private institute. The germplasm pool is nothing but it's a mixture of the wild species and the cultivated species, and based on the mandatory crops which they handle and the gene or trait of interest, the germplasm pool will be maintained by the institute, public institute or the private institute. And finally, the transgenic technology is another kind of source of insect resistance, where we deploy the genetic engineering, isolating the entomopathogenic protein from the microorganism and incorporating into the plant for conferring the insect resistance. So these are the various sources which we use for the insect resistant breeding. And of course, I'll quickly touch upon the wild species of the cultivated crops, but I'll be mostly restricting to the cotton because of our expertise for the past 48 years. I'll be touching upon only on the cotton, but just I'll throw some light on the wild species. These are the few wild species which are used in the insect resistant breeding: cotton, rice, maize, and soybean. You can just go through the few wild species. A lot of wild species are available. Still, I'm touching upon only the widely used insect resistant wild species in these 4 crops. And is it visible, this one? And coming to cotton wild species, the major sucking pest tolerant wild species are from Africa species. Anomolum, longicalyx, this herbaceum, all these wild species from Africa we use for the sucking pest tolerance. And the arboreum is our desi cotton species, our native species of India, Gossipum arboreum. And these two are not so much of economic importance. And the wild species of G. hirsutum. The widely cultivated commercial species is G. hirsutum. And the origin of the species is from Central America, G. hirsutum. G. hirsutum, among the 4 major cultivated species, first G. hirsutum ranks first, followed by the herbaceum, followed by barbadans and arboreum. Now, these are the 4 widely cultivated. Among the widely covered species is G. hirsutum, and origin of species is the wild species is from Central. These are all the various maize cultivated. The entire wild species maize cultivars are from North, Central, and South Africa. And rice, of course, no doubt, the wild relatives are from India and South Asia. Very few species from Africa and Australia. And soya, most of wild relatives confined to Australia and very few around China, only 2. Otherwise, most of the wild species are from Australia. So coming to the plant defense mechanism, the insect resistant breeding, these 4 terminologies, again, which we studied in our host plant resistant: anti-zoonosis, antibiosis, tolerance, and escape. So this the magic of these 4 terminologies I have witnessed in the seed industry. God is very kind enough to me, and I have seen for the past 23 years the magic of these 4 terminologies: anti-zoonosis, antibiosis, the tolerance, and escape. So you all know very well anti-zoonosis. It is mostly related to the morphological characters. So the host undesirable to the insect for the food, shelter, and the reproduction. The antibiosis, adverse effect of the host against the reproduction and growth of the insect. And the tolerance, in spite of heavy attack, even after the reaching the economic threshold level, the plant or the variety or the host or the hybrid able to produce the desirable yield is tolerance, which usually the breeders, the product developers usually rely on tolerance rather than the resistance. And the escapism is nothing but skipping the season or bringing the early hybrid, so that which skips the pest cycle. We all know. Even our the recent studies, the influence of the weather parameters for the sucking pest against maize, we collated, we critically analyzed, and finally we recommended to few of the varieties, susceptible varieties, but high yielding varieties. Few high yielding varieties are sucking pest varieties, which we have found in the maize cultivars. So we advise that not to cultivate during the summer season because of the aphid incidents. Likewise, the escape mechanism which we use to avoid the pest cycle or avoid the plant injury. This is another fourth mechanism. These four mechanisms altogether build a robust insect resistant breeding program. So coming to the basics, we all know about the resistant injury. Resistant is nothing but a completely immunized plant which avoid the plant injury. To some extent, you can take the BT technology, Cry1 AC, Cry2 AB. After egg laying, the first neonate larvae, when it scraps the leaf, immediately mortality will occur, and completely the plant will be free from the insect damage. So, which usually the 100% resistant. So, the midline of the resistant is the injury. So, before injury, the free from the insect damage is the resistant, and tolerance is that the same level of damage, the same level of the economic threshold level, the plant or the host or hybrid which is able to produce the desirable yield is the tolerance. Usually the product developers or the breeders, they rely and love on the tolerance factors. So the participants who are on the online, please excuse me for a few minutes. So I want to have a dialogue of Tamil, the non-Tamil participants, please excuse. So we can, I can quote Vadivelu's famous dialogue, எவ்வளோ அடிச்சாலும் தாங்குறாங்க, எவ்வளோ அடிச்சாலும் தாங்குறாங்க வந்து ரொம்ப பிடிக்கும். Why? Because even after the heavy sucking pest pressure or heavy pest pressure also, if the variety or the hybrid is tolerant to that sucking pest, then it is a better variety rather than the resistant variety. That's why usually the farmers or the breeders or the product developers rely more on the tolerant hybrid rather than the resistant hybrids. So the எவ்வளோ அடிச்சாலும் தாங்கிறது வந்து and the tolerance level. So this is a drawing line. Injury is the drawing midline, where the left will be the resistant and right will be the tolerance based on the economic threshold. This is the another thing which usually breeders, which they look upon for the insect resistant breeding or insect tolerance breeding. And of course, the non-preference mechanism, antizoonosis, as we all know, non-preference of insects, the various non-preferring character, morphological character. Coming to the bollworms, smooth leaves, nectarless, hard bolled rind, okra leaf, and long pedicel all non-prefer. Bollworms do not prefer this nectarless hybrids, or less preferred hybrids or varieties will be the smooth leaf hybrid or hard bolled rinds or long pedicel variety or hybrids. The entire presentation, as I said earlier, it would be more of the cotton only, not more of the cotton. It will be 100% towards the cotton only. So boll weevil is not a major pest here, but it is a major pest in US cotton growing tracts. So boll weevil, the non-preference mechanism of boll weevil is frigobract and red plant, which I will be taking in the next slide. And we at India and South Asia, we concentrate more on the jacid and whitefly tolerance. So non-preference mechanism of jacid and whitefly tolerance is almost reverse. Jacid, as you all know very well, jacid non-preference mechanism is hairy and leathery, and whereas whitefly, it is glabrous, smooth and succulent. So we concentrate more on the jacid and whitefly tolerance or resistant mechanism in our cotton varieties or hybrids. And bollworm, as we all know, transgenic technology, BT is the most successful the insect resistant program deployed in cotton. And these are the few pictures, the glabrous pictures, the glabrous non-hairy, hairy, which we consider for whitefly and aphid. The thick rind is non-preferred morphological character, antizoonotic character for bollworms, and thin rind is susceptible variety or hybrid. Pink plant is a kind of plant in Gossypium hirsutum, which we use for the insect resistant breeding, a purple wash or pink wash, red wash kind of plant, which is not a cultivated species. Usually cultivated species will be of a green plant, green wash structure. But for the insect resistant breeding, for our various trait or gene of interest, we use this red plant breeding. Usually we in the rice also the purple rice which we use in the, and again the thin leaf and the thick leaf, the morphological characters. As I said, the Frigo Brath at the early reproductive stages, in comparison with the normal Brath, which in US growing tract they use for insect tolerance breeding for boll weevil, which is a major pest in various countries. And this is the boll at the boll formation stage. This is 3 weeks boll. 3 weeks boll, Frigo Brath versus normal Brath. And this is a larger interest thing, which usually we depend on the insect tolerance or insect resistant breeding. Post BT introduction, we all, both public and private, we work on the sucking pest tolerance in cotton, and mostly we depend on this trichomes from trichopore. From single trichopore, if it is a single trichome will emerge. In a few varieties of germplasm, in single micropore, multiple or tuft of trichomes will. This is another zoonotic. All the characters, all the anti-zoonotic character or morphological characters are widely used in the insect tolerance and insect resistant breeding. So this one kind of hairiness for hairiness. And in hirsutum, various trichomes, usually it will be deployed for the breeding programs. Type 1A, low density of singular trichomes, and 1B, high density of singular trichome, a sparse trichome, a dense trichome. So these two trichomes have a very good resistance. It confers resistance to both jacids and thrips and aphids. And this is one of the best source for the high resistant jacid, thrips and aphid. What is called pilose type, very high dense, very thick trichome, very long trichome. But the problem with this trichome is it is highly resistant to the jacids and thrips, but highly susceptible to the whiteflies. Few years back, we had a practical experience of this kind of variety where with a dense pilose type of trichomes, up to 80 days, the variety was stay green, completely free from the sucking pest, even below the economic threshold level. The comparative varieties, we have seen the higher ETL level, but the very next plot, very low pest population. But after 90 days, unusually, in that agroclimatic condition, usually that agroclimatic condition where we tested, it was the whitefly low population agroclimatic. Unusually that year, because of the high humid weather condition, whitefly infestation started and completely devastated. Within 4 days of observation, completely this high resistant variety of jacid and thrips turned into a dark black field because of the black sooty mold fungus of the whitefly infestation. So always we have to play a mediocre role or a moderate kind of thing with a sparse or a medium trichome density, which can provide a multiple resistance to the sucking pest. So barbadans, this is a glabrous type which usually adapted for a whitefly resistant. The glabrous, completely non-airy, smooth. These pictures are the stem pictures. Usually stem pictures and the leaf pictures will be of a similar character. These are all the stem pictures, the magnified stem pictures for demonstration. So glabrous type is non-hairy type. and the sparsely grown tuft in the same Barbadans germplasm. These are the sparsely grown tuft and extremely short. Earlier I have shown high dense, a long trichome. Here extremely very short, minute trichomes, but very dense trichomes. These are all the various the variations among the various species and the various germplasm which are used for the insect tolerance or insect resistant breeding. So this is the second, the third thing is the nectar glands. Nectar glands, the research work on nectar glands started somewhere in 70s, it seems. Still it is on the process. Still we have to get a breakthrough of nectarless against the bollworms. So nectar is nothing but, you all know that it is a honey gland. It is an exudate of a phloem. So which usually a sugar concentration consists of glucose, fructose, and amino acids, which attracts the moths for the feeding or for the ovulation. So this nectar glands is a preferred character for the moths and the Leptopteris plus. So adapting the a lot of nectarless accessions are available in our country also. In each species we have almost 40 plus accession for nectar glands. So the scientists right from 70s, they are working on bringing the resistance or using the nectarless hybrids for pink bollworm, especially for pink bollworm, usually for all bolls bollworm and especially for the pink bollworm. So so nectar glands, there are a few, it depends upon the variety. Nectar glands may be the floral nectar gland or extra floral nectar glands inside the floral structure or outside the floral structure in bracts or in the petioles or in the pedicels. So this is the floral extra floral nectar glands. You can see difference, devoid of nectar glands and the nectar glands in the fruiting bodies at the base of the calyx. This is another character, morphological character which we deploy for. And I usually what I said, even in leaf, you can see, you all might have witnessed this nectar. Usually in all varieties, this nectar glands are available. This lower portion of the leaf, as the maturity depends upon the maturity, this nectar glands will turn blackish at the later reproductive stage. And the nectar glands usually depends on the variety, germplasm. Even in few varieties, the nectar glands are visible only during the anthesis period, and few varieties it is visible only during the early reproductive period. Like that, it differs upon the variety, the visibility of the nectar glands and in comparison with the non-nectar glands. So usually for breeding against bollworms, we use nectarless hybrids. So a lot of accessions are available globally and also in our country for utilizing this. And as I said earlier, glabrous for whitefly, hairy for thrips and jassids. But what we can use for the multiple sucking pest tolerance in Gossypium arboreum, there is a unique character wherein the phloem is deep below the because of the long palisade cells, it is placed deep below so that the mandibular section of the sucking pest cannot reach. So in Gossypium arboreum, it can confer resistance to all three sucking pests or all four sucking pests, aphid, jacid, thrips, and whitefly, because of the leaf arrangements. The novel leaf arrangement hinders the sucking pest to reach the phloem, and the traits from the wild species or Gossypium arboreum is transferred to Hirsutum or the Barbadans through wide hybridization for conferring the multiple sucking pest tolerance. So this is all about anti-zoonotic characters which has been exploited or which has been commercially and successfully utilized for insect tolerance breeding. And coming to antibiosis, the biochemical characters, high dosical content cultivars used for bollworm and mite resistant, phenol content for bollworms, and the high content tannins for aphid and spider mite, and leaf tannins for the JASIC tolerance. This is the second part of mechanism of resistant, which has been widely used for the plant defense mechanism, utilizing the plant defense mechanism for the breeding antibiotics. So this earlier work attempted on the interspecific hybridization using the wild cultivars paved way for the current generation development of the varieties and hybrids. This I can forward to you the various work just for the reference. Even the resistant to the, just as I said earlier, Rhizotum with Anomolum and Barbadans wild species. And for Thrips arboreum and Rhizotum arboreum. So with all this, after studying all these antizoonotic antibiotic factors, now the breeders will select the right breeding method to introgress the gene of interest or the trait of interest. So various breeding method, which again I apologize, I can't do justice of explaining this breeding method. Only breeder can do. Right. So, but on a broader aspect, I can say that the 5 breeding methods used for insect resistant or tolerant breeding is the pedigree breeding for new line development, male female crossing, and the line development F1 crossing. And the second is the back crossing with the integration of oligogenes to maintain the isogenic lines. And third is the wide hybridization, as I said, with the cultivated species with the wild species for the transferring the trait of interest. And transgenic technology, we all know the BT technology, which is commercially cultivated in India. Almost 95% of the cotton cultivated area is under BT technology. And now the cutting edge technology, the disruptive technology, is the genetic engineering technology, that is RNA interference technology and the CRISPR-Cas technology. RNA interference technology, which I will be taking at the later, still it is in the nascent stage, and only few commercial products are released or launched, which we will discuss. This, as we all know, the protein isolated from the Bacillus thuringiensis, the crystallized protein, insecticidal toxin, which binds. This is the mode of action of BT technology, which binds to the midgut epithelial cells and causes paralysis and death of the insect, and widely used for the Lepidopterus pest resistant, and most successful are the BT technology. The BT cotton, which widely we use in country. These are all the various insect resistant technology commercially available in India. Only the Bolgar 2 technology, Cry1AC and Cry2AB, is still available in India, and remaining all it is under the regulatory purview and still not commercially launched. And the gene responsible for insect resistant and the developer and the technology provider I have listed out here, the Monsanto and the Cry1AC genetics, Metalix, and the Cry1AF with VIP3. VIP3 is a Syngenta's proprietary gene with Dow AgroSciences. And the Syngenta VIPCAR gene, VIP3 is another gene which is widely used other than India for insect resistant transgenic technology. And white stripe is also another widely adopted, the commercially launched technology, not in India. And as I said, the RNA interference is regulating the gene expression in vitro and in vivo, and only again and again, as I'm insisting more of biotechnology and breeder, but still it is a gene silencing mechanism. It may be the plant mediated RNA interference through host plant the insect, the target insect, the gene may be silenced and the target pest may be controlled. And Colorado potato beetle and Asian corn borer, against these two pests, the host plant mediated RNA interference has been successfully working. That is what the report says. And commercially, for Western corn root borer, Bayer has launched technology which is called as a plastomic plant with high RNA efficiency, that is commercially launched against Western corn root. That is one type of the adaptation of the technology. Other type is the RNA sprays, direct RNA sprays which silence the growth and development of insects, which is also happening in the corn rootworm and locust in various countries. This RNA direct sprays towards the insect, that is a cutting edge technology which is still in the nascent stage. One is the plant mediated RNA technology. Second is the direct spray RNA spray technology. And this is going to be the next generation insect resistant technology in future for protecting the crops. Usually now it is majorly concentrated towards the sucking pest of various crops. So coming to the durability of insects resistance, we all know we have some limitations because compared to the crop life cycle, the number of generations is more for insect. For example, PBW, in 150 days of crop duration in Central and South India, it has more than 6 to 8 generations, whereas in North India, the generations are very low due to the overwintering diapause. Controlling pink bollworm in Central and South India is challenging compared to North India. So that the resistance, the continuous, the pressure, the selection pressure of the insect, and the short duration, maybe the short duration compared to the generations, the durability of resistance becomes questionable due to the adaptation of the insect. And the second is the boom and burst cycle of the both the insect population and the variety or hybrid. Boom and burst cycle, usually we introduce, or the breeders or the technology provider introduce a variety, insect resistant variety. That is called as a boom cycle, wherein we will have a very good insect pest management. Over the years, or the acreage, increase in the acreage slowly due to the selection pressure or due to the mutation or due to the resistant development of the mutant population of the insect, suddenly it arrives at decline phase. Which arrives the decline phase? The resistant or tolerant varieties reach a decline phase, which is called the bust cycle. Again, a breeder or the biotechnological intervention or any other intervention will happen to revive the same variety or the improvisation of a cultivar or variety, so that the epidemic state of the pest will come down, and again the boom cycle will increase for the insect tolerant variety. So the boom and bust cycle, usually it will be monitored very carefully by the breeders, entomologists, and the field agronomists very carefully. If it is a conventional breeding, usually they will take 3 or 4 years before they will try to add or replace the hybrid. Usually we started with RCH2 for Central India, then 659, then now we have a acid tolerant hybrid called Rasi Magic and Rasi Magna kind of thing. Like that, we usually replace the hybrid to maintain this boom cycle, so that the bust cycle should not affect the farming community. That is the hidden agenda behind the boom and bust cycle. And unfortunately, the same thing happened for BT technology. Insect resistant management studies, entomologists, IRM, all They have monitored very early during 15 and 16, Cry1 AC and Cry2 AB. It is going to lose its teeth against pink bollworm, being a monophagous pest. They all alarmed the pink bollworm will develop resistance very shortly. Unfortunately, due to the anti-science activists or due to some regulatory issues, we have not done any pyramiding of genes. Now the pink bollworm population is 100% resistant population in country against bollworm. Now the BT technology against pink bollworm is in must cycle. We all enjoyed since 2002 the boom cycle, the plastic kind of plant, no Lepidopterus pest, no larval, only a small 250 ml of sucking pest molecules is sprayed. But now the time has come. Now the farmers have started to give 4 sprays of synthetic pyrethroid or organophosphate or the new generation CTPR kind of Corogen or the spinosad for controlling pink bollworm, because the non-compliance of refugia because of the regulatory issue in pyramiding of genes to revive the technology, it all hindered, and now the farmers are facing the age-old non-BT era. Of course, not like Helicobacter. At least now they have started spraying of food. So this is what the boom and bust cycle. This is where the 100% resistant, the durability of resistant play. That is why I always quote that tolerance is better in spite of hitting several times. Still, it gives the desirable yield, the tolerance hybrid, but the resistant hybrid suddenly, if not monitored properly, if not upgraded properly, it will go to the bus. So again, the third reason is the durability of the resistance depend upon the continuous cropping. Maximum chances of the insects to boom, the continuous cropping without any crop rotation, or after harvest, not removing any of the crop residues that which harbors the pupa or the continuation of the life cycle and all will help to boom the insect cycle. And so usually in conventional breeding, what the breeders used to do is that they used to shift this horizontal vertical resistant to horizontal resistant instead of the oligo genes of few genes holding the resistant. They concentrate more on the polygene, that is the horizontal resistant. And now we all started, because of non-compliance of refugia. Now government has advised all seed companies to give the refugia in bag. Earlier we used to give 120 grams of non-BT, which usually the farmers will not cultivate, they throw away. And due to high dose refugia strategy in Gujarat, 1 in a million population, usually resistant population occurs in any of the organization. Similarly, 1 in a million resistant population, 1 male emerged in Gujarat, 1 female emerged in Gujarat in the same area, both resistant population mated. 400 eggs fecundity rate, 400 into 400. Now, within 3 to 4 years, 100% pink bollworm resistant population. So, what government said is to mix the non-BT isogenic. Isogenic means the same variety. If it is RCH2, same RCH2 variety, 25 grams should be mixed in the 450 grams packet, so that the farmer cannot differentiate, and usually it helps to extend the shelf life and by reducing the resistant insect resistant. So this is already I have explained. This is a pictorial representation of boom and bust cycle. The breeder produces the new resistant variety or tolerant variety. The crop increases, the acreage increases. Suddenly insect become the epidemic, and suddenly the susceptible variety crop decreases. The pest population increases. Again, an intervention has to happen through the conventional breeding or through gene pyramiding. Then the boom cycle will start, pest population will. So this has to be monitored, and this has to be carefully intervened at the regular intervals. So the limitations of the insect tolerance breeding, as I said, the resistance of the insects are very smart organism, and interspecific gene transfer, genetic genes transfer from wide hybridization. Sometimes the unwanted things may carry on to the offsprings, like any kind of disease, that we need to be very careful. The long-term process, as I said, this is not a very easy process. It is not a chemical preparation process. It usually the pre-breeding program, selection, line improvement, breeders toil in the field throughout the year, and for releasing, they have to one variety, they have to screen thousands of variety, then from 1,000 better performing variety to the next stage. There again the producibility, all this, it will take minimum of 8 to 10 years for releasing one insect tolerant or insect resistant variety. That is the very normal process. So it's a long-term process. Again, as I said, the pyramiding of genes, all that regulatory legal issue, all these issues in bringing the novel technologies. Now anti-science is more active than the science and academic community. That is another bottleneck in bringing the novel technologies. So very quickly, this already I have explained. So I will come to the commercial part of this insect tolerance breeding. Apart from the yield-centric character or a triad required for a farmer or a customer, the additional triad, which is very, very important, is the insect tolerant or insect resistant breeding. So usually in our country, we concentrate, because of the BT technology, we concentrate more on the sucking pest technology, Jacid, Thrips. So as I already explained, the hairiness, leaf thickness, and tolerance level. For whitefly, the smoothy, small, erect, and less foliage. So these are all the various triads. You can see a clear-cut difference. That is what I say. God is kind enough me. Whatever I learned in this eminent entomology department, God has given me the opportunity to see that everything for the past 20 years in the field. The same 4 rows of variety in the screening program. You can see the devastated susceptible line in the very thing which we will remove out of the product basket. These are all the things susceptible. You can visibly see in the very next row the susceptible line. So these are all the characters which we usually pinch upon: the thick leaf, hairy leaf, hairy stem, and hairy weather. Usually screening happen in the hotspot areas, open field conditions. So a lot of grading things happens. We have grade 1, 2, 3, 4 for grading this insect damage symptoms apart from the population level. So for the simplified grading method, all the validated standard operating protocol we usually follow for insect resistant. So thrips tolerance. Thrips, another problem is thrips. Thrips nowadays, it is becoming a very big menace to the cotton. Because it attacks even the first true leaf stage, immediately at the cotyledon stage. Some absentee landlords who visit after 20 days, they say the germination problem. If you go and diagnose the germination would have been 100%, but the early thrips incidents would have completely devastated the crop. So the thrips, even the dry and humid condition, controlling of thrips, management of thrips is a very big challenge. In dry soil condition, as you all entomologists know very well, it reproduces, rightly said by Claire, it 7x times it reproduces very quickly. Even after spraying, within 3 to 4 days, the pupae will attack the plant and create. And very few thrippicide are available. All other older generation thrippicide lost its seed. The newer generation being expensive, it is not widely used by the farmer. So the solution will be the insect tolerance lines, cultivar varieties. So this is the early stage, the thrips infestation susceptible line versus the tolerance line. And still 70 days, the thrips incidence will be there. This is at the time of the early reproductive stage. The tolerance line versus susceptible line. You can see the grenishness, the crop figure. So the thrips incidence field will lose its vigor, losing the vigor at the early stage. Straightly, it will cut off 15 to 20% of the yield, affecting the yield attributes like monopodium and podium. And another all good real pictures captured from the field, difference between the susceptible and tolerant line, and the triads under consideration are thin leaf, wavy leaf, rectangular leaf, sparse medium stem, less foliage. Less foliage is another big triad which usually, and this CLCV is a very big issue in Punjab, Haryana, Rajasthan, and usually this screening, all kind of screening, absolutely it happens only in the hotspot areas. That is where the entomologists play a critical role in identifying the hotspot, the pest life cycle, when will be the peak population, when will be the peak population in the agroclimatic, then they will pass on the information to the breeders, and it will be helpful for the breeder to conduct effective screening studies for bringing tolerant practices. So achievements of the public sector in the insect resistant breeding for Jacid tolerance. These are the few resistant and tolerant varieties published by the public sector. Whitefly, Kancana, excuse me. Excuse me. So aphid tolerant and MCU5. MCU5 is not only the stem weevil tolerant variety, it is also a verticillium tolerant. Recent MCU5 wheat is a verticillium. And the achievements of Rasi seed, the 48-49 year, very strong market leader and cotton leader, envisioned by agriculture graduate Dr. Ramsamy from a small town called Arthur. Now it is a household name for the each and every cotton farmer in our country. This Rasi seed, so undisputed leader in the cotton, and these are all the North Indian varieties, Jacid and Trips tolerance, 602, 650, 653, and the new launch 926. And these are the most demanded hybrids in North India, Punjab, Haryana, Rajasthan. The second, the big portfolio is 773, 776, 926, only because of, not only, I'm sorry, it is only because of yield, and second to the yield, whitefly and CLCV tolerance. 8 years before whitefly devastated, wide, huge outbreak, and after that, the launch of these three hybrid gained importance. Believe me or not, now these three hybrid constitute 50% of the market share in North India. 5 out of 10 farmers cultivate these three hybrids in North India. Even if you go and visit few of the villages, almost 7 to 8 farmers out of 10 farmers, they cultivate either one of the three hybrids. All these are achievements of the breeders and bringing the insect tolerance along with the high yielding triad with a very good cotton quality. And the central and southern India hybrids are the 779 Rasi Magic, RCH 797, RCH 608, Jacids and Trip tolerance hybrid. Magic and 659. 659 is the widely cultivated, extensively cultivated hybrid across central and south India. And as I said earlier, tolerance level. Early stages, this would be Jacid susceptible. But after the boll formation, the farmer used to love this hybrid. That is why this hybrid is cultivated across central and south India. This is Jacid susceptible, whitefly tolerance. Of course, there is no much whitefly incidence in central and south India like North India. Though it is Jacid susceptible, because of its tolerance, revival, reflushing capacity and high yielding capacity, it is still loved by central and south India cotton farmers. So these are all the various achievements of the insect tolerance hybrid, and we always love to associate with academics department and universities with a long-term goal and objective, working together to bring more smiles to the farmer. And I take this opportunity to thank organizers, professors, graduates, and online participants for giving me the opportunity. Thank you. Thank you. Thanks.
I request the participants, those who are online, please post your questions in the chat box so that we can start the interaction session. So those audience who have been physically present here can post, then can ask the question directly to Dr. Suresh Kumar. The Indian move on the customer. Chat open. So Dr. Ramesh Kumar, you can post your question, or I'll unmute your mic. Ramesh Kumar, mic.
Dr. Suresh, we thank you for your elaborate lecture on resistance breeding for insect tolerance. And I have one doubt, that is, how about combining breeding for agronomic traits to supplement or to complement the resistance character in the croplands?
Yeah. Thanks, Ramesh. Yeah, already that is under not under consideration. That is, breeders are on that project, and they are successful on the project to improve the germination and the earlier vigor, which can support the resistant factor. So I have not covered all that, the agronomical factor and the quality parameters, but it is already in progress, the agronomical characters which can support insect tolerance.
Yes, the most important factor that brings in resistance to Bt toxin is the duration of the crop in as far as cotton is concerned.
Yeah.
So in that case, how about bringing the crop duration will help developing resistance?
Yeah. The earliness compact hybrids already the breeders are working on that factor, and very soon the hybrids will be launched on that factor, the early duration hybrids.
Thank you.
Thank you.
This BT cotton work for managing spot after.
Yes, BT cotton, the early instar of BT cotton will work against Spodoptera altera. But any migratory, the voracious third or fourth instar from the adjoining pulses field or soya field, the efficacy will be low. The BT cotton works against all early instar, both the leaf feeders or the borers or the bollworms. It works well against the leaf feeders. Any migratory Spodoptera, the efficacy will be low. Migratory late instar larvae.
So the nectarless varieties are preferred for pest
No, I
resistance.
can. No,
But,
no, sorry.
I think it will be difficult for you because of your age. It is nice for you, then it's okay.
No issues. So this glasses can support. The nectarless varieties are preferred for pest resistance, but many of the literature say the cotton varieties which bear an extrafloral nectaris actually give protection to the plants by way of attracting several natural enemies. Yeah. This is a good question. Yeah, this is again a debatable topic. This since 70s it is happening. Yeah, good question. Even the seed production, seed growers, they said that we want nectar hybrids. Why? Because the seed production will be more in nectar hybrids. But seeing the challenges in controlling pink bollworm, still it is in exploratory stage only, though this nectarless hybrid again bollworm. But as you rightly pointed out, against natural enemies and the seed growers, they prefer nectar hybrids.
But in field level, we have observed.
Yeah, it's a truth.
Epidopteran pests were actually trapped by the ants.
Mm.
Because of the extrafloral nectaris, they get attracted to the ants. The ants are controlling the Epidopteran pests. We have recorded it. She is actually an experiment.
Okay.
That's why she goes.
And moreover, still we are exploring the nectarless characters against bollworm. If it is a very successful thing, we would have been launched a lot of varieties, and still it gives a very minimal protection against bollworms. So still we are breeders are working on this. It is not so successful like the transgenic technology. Still we are exploring that nectar. That's what. Yeah, that is the reason I have quoted along with our thesis on the different agroclimatic zone, different pest pressure will be there. So the Jacet tolerance and thrips tolerance will be more in the south and central area. So whitefly and CLCV, the pressure will be more in North India. So we accordingly we concentrate. Even all crops we concentrate like that. Even as I said, the summer maize, the aphid incidence in few cultivars, so better we will avoid that hybrids or varieties. So it depends. The basic studies will support for bringing out the final product. Yeah, that is again the long process which I said, the multiple, the resistant from the wild species through wild hybridization, we can bring, but we have achieved that and still we are working on it. Still the scientists and researchers are working on the multiple resistant factors. Okay. Yeah, it's a kind, it is a one more mechanism of pest management. The earliness, for example, ping bollworm now, what the scientists and researchers say, let us being rightly pointed out by Dr. Ramesh Kumar also, the earliness of hybrid will skip 1 or 2 generation and will minimize the yield loss. So that is one kind of mechanism. Cutting down the duration of the life cycle of a crop. So that is why I've included that escape. That right now it is followed in the system. Age 5. Yeah, it is working. That we it has to be go through a community approach. This mating disruption technology yesterday and today also our guest has presented. Mating disruption technology will work. It is again a disruptive technology. It works well, and it has to be followed as a community approach in large acreages. This mating disruption technology, it works. Sir, I need a separate half-day presentation for that, sir, to break the myth and reality of breeding. That is where the vocal nature of anti-science activity, activists has created even that fear mongering, even to within the academic, even within the scientists. So, in a single word, it is a beneficial thing which has to be launched. But this is not the forum to discuss. But if you need, please give me half-day time to break that myth and realities. How much biochemicals in insect resistant in BT cotton? Only very few. What I said, high gossipal, high tannin content. This is what not, it is not exploited as like the anti-zoonosis. What is the status of maize insect resistant breeding by RASI? I'll connect my RASI maize breeder. It is they are working on it. For more details, I'll connect with RASI maize team. If changes some character in particular crop, that change will create impact on another crop to adapt host plant of the insect. No, I'm not exactly. Any thrips tolerance hybrid released in vegetable crops? Yeah, very few hybrids released. I'll send you the list which is available. I'm not very sure on that aspect. I'll provide you, but few the thrips tolerance varieties or hybrids are available, but not resistant, because chilies or the thrips and mites, the infestation will be very high in chilies usually, but very few resistant, not resistant tolerance varieties are available. Success rate of RNA, only one commercially RNA product against Western corn rootworm by Bayer, and the plant mediated, it is the researchers say it is working well against the potato Colorado beetle and one more Asian corn borer. So these three are the good examples which I can say for. So Kannan is online. So, okay. Morphological character and biochemical character, which one is, both are very important. So which one is easy to adapt, we are adapting, and both are important. All the four, the magical terminologies which I mentioned are very much important for giving the insect tolerant to breeding. So we can't say, we can't prioritize anything. Is there any possibility that use of BT cotton usage lead to outbreak of any secondary pest from Coleoptera family? To my knowledge, because of introduction of BT, I have not observed any Coleoptera pest in BT cotton field. If there is any scientific evidence, please let me know. Let me go through it. Yeah, Kannan. Kannan. Kannan, am I audible? Kannan, good evening, Kannan.
Yes, no.
So the field level BT protein detection, can you just brief out BT protein detection kits in at field level?
That basic primer already designed by Central Institute of Cotton Research in Nagpur, and some few private companies also. Stick formed this primers coated sticks available. We need to just take young leaf, 1 rupee coin size leaf, and just crush it, and that we need to put that crushed solution in that stick. If BT content, BT protein content available in that leaf, it shows some band, this pink color band. This already this kits available in market. In CSR also they have.
The field kits are available for BT protein testing at field level, the leaf tissue kits. Any other question? So thank you all.
I request Dr. Nalini to propose formal vote of thanks to Dr. R.S. Suresh Kumar.
Dr. Suresh, on behalf of IQUAD 21 team, I immensely thank you for your lively, detailed, free-flow, lucid presentation on insect resistance breeding, starting from source of insect resistance, plant defense mechanism, insect resistance versus tolerance, non-preference mechanism in cotton, and you have shown very clear photographs of different trichome types versus resistance to insects, which will be a greater help for better understanding in students. Also, you have thrown light on nectary glands, antibiosis, advanced breeding methods like direct RNA spray. Thank you once again for sharing the minute details from your vast experience from field with great patience, and also enlightened us about the present scenario going on in cotton pink bollworm, thrips, and the greater efforts by the breeders in pest management. As you quoted, let us together work to bring more smiles to farmers through IQUAD. Thank you. Thank you once again.
Thank you, Dr. Nalini. And we are fortunate to have our professors and Dr. V. Selvanarayanan, Controller of Examinations, with us today in this evening. And I request Professor V. Selvanarayanan, Controller of Examinations, to have some closing remarks about IQUAD 21. Please, sir.
Respected head of the department, distinguished colleagues from the Department of Entomology as well as from other departments, invited speaker, our prestigious alumnus, Dr. Suresh, organizers of this great event, participants online as well as offline. A very good evening to one and all. It gives me immense pleasure, privilege, and pride to be part of this event, as well as to be part of this great department. Well, when I was just listening to Dr. Suresh lecture, I just went back some 30, 40 years ago when we were students in the same department. Such facilities were lacking at that time. The title of the event, IQUAT, Institute, Industry, Interaction, Interface, and again international. You can see the galaxy of speakers right from day one, where Dr. Bhairappa started with pheromones and then about fumigation or regulatory issues or about host plant resistance. Right. As I used to always say, the word entomology can be simply replaced in our faculty as well as in other institutions with the term excellence. Always we excel. We are proud to say that. Hope my extension colleague may permit me to say this. Because I used to always say this to our students also. We deal with living creatures. If you take a breeder, he deals with plants. If we take entomologists, we deal with insects, we deal with the plants, and also we are getting involved. So we need to manage many things. And that's why we are excelling in many spheres. And our department is having a rich legacy of taking up the lead. And as the title of this international workshop highlights, changing contours. When the horizons are expanding, we are the first to step into the new horizon. And when there is a change, paradigm shift in a concept of research, we are the pioneers to enter it. Right, again, I just go back some 40 years ago. We were the pioneers in the country to talk about induced resistance when the topic was not at all discussed in the entire world. Role of potash, the first national conference was organized in our department as a pioneering event where they proved that potash enhances resistance, what we talk about as induced resistance now. This is the rich legacy we have. And during those times also, we took up all the early initiatives, whether it is going to be host plant resistance or otherwise on biocontrol or any other sphere, we just expand ourselves. And this is again another milestone in the Department of Entomology. So I really appreciate the organizing committee, including the head of the department, because we know how difficult it is to get in touch with such a galaxy of industry people. They are topmost people. And again, I don't know how Suresh find time today to come here. They'll be traveling a lot. They have too much commitments. In spite of that, they were so interested to be here to interact with us. So in that way, this is a great event. I really appreciate the organizing committee for the excellent efforts. And to the participants, both offline and online, my request will be the IQUAT, the interaction interface, is not going to conclude with this plenary session. It is going to be an everlasting interaction between you with that of the institutions and the industry. This is what is actually the need of the day. Gone are the days when we were talking about lab to land, when our extension people will be carrying the technology from the lab to the land. But now the industry is playing a major role, and industry is just interacting with the institutions greatly. And this should be the platform for all of you to just get in touch with the respective industry. If you are working on host plant resistance, get in touch with those industry people who are pioneers. Like that, accordingly, you just get yourselves interacted, and that is going to help you a lot. And that is going to be the fruit of this 2 days effort. It is not going to end today, as I said. Just let this be everlasting. So with these few closing remarks, I thank the head of the department and the organizing committee for giving me this great opportunity. Thank you, one and all. Wish you very good luck. Thank you.
Thank you, sir, for your encouraging words. The memories are flowing back in my mind. The day in which I sat in your class as an undergraduate student, I was the day where I got motivated to become an entomologist. Thank you for being here with us today. And I'm also fortunate to have Dr. S. Manikvasagam, Professor of Entomology, with us today. I invite Sir to have some remarks, closing remarks, please, sir.
A very good evening to one and all, both offline and online. It's my pleasure to say a few words about the contours, the changing contours in pest management. When Selva told me, sir, this is the one which we are organizing, and when I was going through the program, I was able to see some missing points, especially the molecular tools in pest management. That is what is the lead of the day. And that was amply filled up by our alumnus, Dr. Suresh Kumar. He even coined the word RNA interference technology, gene pyramiding, and all those things. So that was actually proposed by the real molecular biologist. And really, it is heartening to note, especially by a teacher like me, to see students of our own to mold like that, and then they are now recognized as international scientists, both in the academic side as well as in the industry side. It is really heartening. Hearty congratulations to Suresh Kumar. And we could feel that our students are molding. That's what my purpose of talking this is for you people. It is for you to take such challenges. Okay. Even when Dr. Selva was telling, the others, Dr. Rees was telling, I even go back when I was asked to join here in January 1994. At that time, the whole Chidambaram was flooded in January. I was wondering what to do here. And when I entered here, I could see a big banner on FAO trainers training program on IPM. That was the first training program in Asia. In fact, I can say. So that was again another legacy by our own department. Even Dr. Hari Prasad, he was one of the participants. Everywhere it is raining. Dr. Peter Kenmore, all of you might be knowing, one of the well-known IPM specialists. And he was the, whether it is raining or hot sun, he will not leave you as such. So all the students, I mean participants were in the field, taking an umbrella and then doing all the work and things like that. So when even you might be knowing at that time, plant protection advisor to Government of India, Dr. Raghunathan, he was instrumental in giving the training program to our own department. And it was really heartening to note such events. Even other people are envying that such events are taking place here. Okay, anyway, it is really up to us to hold our flags very high. Unless we are going to hold it high, then who else is going to come and hold it for us? Am I right? It is really heartening. And I really congratulate Dr. Selvamutu Kumaran for conceiving this idea first. And then that was ably guided by our head of department. And I really feel that such more programs should come. And then that should be handled by youngsters, because it is going to be ruled by youngsters in the coming years. And especially the students, you should take up such leads. Even I was thinking what I have done all these years, say for example, for last 28 years. And when I was thinking of Corona, that taught me a lot of lessons. I should have stopped this taxonomic work in between, and I should have gone for these parasites using herbivore-induced plant volatiles. That's the heart of the day. How we should have done this herbivore-induced plant volatiles, and then that should have been something like a pheromone or chiromone that we can think of, and then we can spray that. And then, as he was today afternoon, he was explaining, am I right? The first lecture today. So somebody was asking that question again. So that would have been a nice idea. And again, that was again proposed in the FIST, in the proposed program what we have given. If everything materializes, then that will be again a boon to Indian farming community. So definitely we are not second to anybody. So it is up to us to take it up these challenges. And then I finally congratulate again the organizers and the head of the department for taking up such a nice international workshop on changing contours in pest management. Thank you so much for giving this opportunity.
Thank you, sir. And it is because of the motivating words we have been working for long. And I take this opportunity to invite our Professor and Head to present his closing remarks. Before actually leaving the mic to our Professor and Head, probably all of you would have seen this. For the past 2 days, it is meant business here, right? It is all because of these 3 professors. We have learned what we have been showing here is only the tip of an iceberg that they have. So without them, we would have been nothing to be here. So I take this opportunity to invite our Professor and Head to share his closing remarks.
Good evening to everyone. The most respected controller, Annamalai University, and the professors and other teaching colleagues from entomology and other departments. Once again, good evening. So the IQUAD 21, it will not end in this year. So it will move even for another 20 or 25 years. So we should see that 21, even up to 35, 36 like that. So actually this idea, even in the inaugural session, I told this idea, it is totally conceived by Dr. Sellamuthu Deputy Director in IQSC. But after seeing that, Dr. Ramesh Kumar, another Deputy Director, very active person, they both the personalities, Deputy Directors, they shaped that particular concept. Actually now we are going to adapt this particular concept throughout the university. All the departments in the university should take up this program. That is the idea behind that. Where actually this is a model. We are running that. We are just conducting this program, and we are saying what are all the problems and what are all the good things which are all coming out from these things. Based on this experience only, we are going to suggest the ideas, moralities, everything to all other departments in our university. So we are waiting that circular is ready. So within another 4 or 5 days, the basic things that should be done in this particular workshop, international workshop, that may be circulated within another few days to all the departments. So all the students in our Annamalai University may be exposed to this kind of interactions. That's the idea. So I have to appreciate Dr. Sellamuthu Kumaran and Dr. Ramesh Kumar for such a nice concept. I think no university in India is having such a type of continuous activity. So we are going to force this activity as a best practice for another 5 years. After 5 years, we have to submit SSR for NAC. In that, this may be the first best practice. So I have to thank Dr. Sellamuthu Kumaran and the team, Dr. Kanagarajan and Kadirvel and all other organizing secretaries and even co-organizers for such a nice model related to IQUAD 21. So related to this activity, so you can see the list of scientists from various countries. Even Dr. Suresh Kumar is here. So if I remember the thesis work of Dr. Suresh Kumar, so he actually did that maize work for about 7 years, I think, 7.5 years. Actually, after 2 years, first 2 years, he was there in Hyderabad and he was looking for the company project and other things. He was working there on, I think, BT maize. Once I visited Hyderabad, so it was just like the US office. So most of the rooms in that and all the facilities, they are just replica related to the office in USA. That sort of facility. So I thought our department should have at least 25% of that particular facility. So we are working for that. So our forefathers, Dr. P. Bhaskaran, Dr. Narayanasamy, and even Dr. Nachiepan sir and others, they taught us. Every person where Dr. Selvanarayanan and Dr. SM sir, we are always keeping the time, and we are always seeing the business. We won't divert ourselves in any other activities. So that's the thing we had even in my early periods during 1994-95, when Dr. Selvamutthu Kumaran, Kanagraj and others, they were students at the time. In all Saturdays and Sundays, myself and Dr. Manikasam sir and VS sir, we were just in the laboratories. No senior professors were there. We were always there in the laboratories, and we were arranging the insights, and we were learning scientific names. I'm not that much strong in scientific name, but VS is having a lot of interest in that. He know the scientific name of all the pests. So for that only, I associated, I learned scientific names, how to remember scientific names and I think from Dr. VS. So we were there almost, except Deepavali and Pongal, most of the days, almost around the year, we were there. It was taught by the forefathers of the department. So I expect such a kind of activity from the students and scholars. So the thing is, just now I discussed with Dr. Kannan and Ayasami. Our aim is Dr. VS and SM sir. SM sir is going to retire within another 1 year. But our efforts, at least within another 10 years, our department should have the tag CAS. You know that CAS, Department of Entomology, Center for Advanced Studies. That status we should achieve. For that, I am planning to create a roadmap for towards that CAS. So you work for 10 years, definitely you can get CAS. That we want to see. Then Selvamutthu Kumar and others are here. So you have to work for that, and you should bring that lorry to our department. So I'm much worried about time. It is almost half an hour excess. So with this few words, I congratulate the team for all organizing this, and I congratulate and I thank the students for all actively participating in this program. Thank you very much.
Thank you, sir. And Dr. Professor Selvanarayanan has some remarks to say.
Not a remark, just a very happy news to share with you. Hope our head of the department will keep everything with him always. Whenever it is going to come for a prize or an appreciation, he used to just shun away. And I have a very happy news, right? One of our scholars in the department, Ms. Suguna, has been awarded with the prestigious Inspire Fellowship. Just now I heard the news from him. When I was sitting with him, just interacting, he was telling me, and he never expressed that, saying that it is not going to be my effort. So this is a first of its kind in the department, I hope, Inspire, from Inspire. So appreciations to the scholar and also the guide. Thank you. Thank you.
Thank you, sir. And I sincerely acknowledge and sincerely assure our process that we will keep the light burning, and we will make the department flying high in the sky of academics. And a friend in need is friend indeed. With this quote, I welcome my best friend and classmate, Dr. C. Kadirvel, the convener of the workshop, to present the workshop report.
Respected controller of examinations, respected head of the department, respected Professor Dr. Manikasam sir, colleagues from other department and entomology, my dear student friends, and the participants, those who are in online. Good evening to everyone. So I take great pleasure to compile and present the whole report of the workshop. So to bridge the institute and industry to have interaction and interface with each other, the IQAD 21 was planned under the initiative of IQAC of Annamalai University. The current topic was proposed to address the problems in IPM, HPR, residue management, and also agrochemical development. This international workshop on changing contours in pest management was open to undergraduate and postgraduate students of Annamalai University and also scholars and researchers from other institutions of our country. 2 technical sessions per day were conducted, in which 9 eminent personalities were participated and delivered the lead lectures on pest management. Out of 9 speakers, 4 from foreign countries like UK, Italy, and Australia. On the day 1, Dr. S. Arivudanambi, Professor and Head Department of Entomology, Director, IQAC, and Advisor, IQAD 21, Annamalai University, welcomed the gathering. And the program was inaugurated by Dr. M. Ganapati, Dean, Faculty of Agriculture, Annamalai University. Dr. Selvamuthukkumaran Tirunavukarasu, Associate Professor, Entomology, and Director, IQAD 21, Annamalai University, has given a brief introduction about the workshop. Dr. B. Ananda Kanesaraja, Organizing Secretary of IQAD 21, introduced the speakers of first session and requested to deliberate over the latest trends in pest management. Dr. Bhairappa, Lead Scientist, Entomology, Gayajan Technologies Private Limited, has given a magnificent presentation on the current status in decoding insects communication. He elaborated his ideas on chemosensory organs like olfactory receptors with Drosophila as a model insect and semiochemicals to monitor and manage the pest. And he elucidated the fact that the sensory receptors in insects are precise to detect few nanogram or picogram of semiochemicals. He presumes that nearly 15% of farmers utilize the pheromone technology, but it is tremendously increasing year by year. He also emphasized the point on the shift from insecticide to green solutions. Dr. S. Ranganathan, CEO, 7-Star Fruits Mahiko Grow Company, Mumbai, has delivered the requisite on residue management in table grapes export. He enumerated the various certification required for export, suitable varieties of grapes in India with its harvesting seasons. He threw light on potential market for grape export, major pest and disease of export concern, residue levels, and various steps involved in the process of grape export. The speakers of the second session was welcomed and introduced by Dr. M. Ramanan, Organizing Secretary of IQAD 21, to share their ideas in insecticide resistant management and newer insecticide development. The topic insecticide resistant management was presented by Dr. Luigi Avella, Global Technical Development Manager, UPL Limited, Italy. He apprised that insects, fungus, and weeds are developing resistance to pesticide around the globe with a faster speed than the discovery and development of new active ingredient. He discussed about the evolution of insecticide resistance in BPH along with examples. He enlightened the tactics followed to avoid the insecticide resistance wise monitoring, application of correct dose, alternate application, tank mix, and IRM and IPM education. Dr. A. Prabhu, Technology Lead, Insecticides, UPL Limited, India, has delivered the lecture on perspectives on newer insecticide development. He emphasized the need for new and safer molecules with new technologies in crop protection. And the new crop protection products should be biologically efficient, user-friendly, environmentally sound, and economically viable. He also elaborated the invention process of the new product and sources of new lead development. He enlisted the insecticides and the development since 2010. He added that Flupyrimine is the product of UPL Limited, is in advanced stage of development for release for the management of rice BPH and OSB, yellow stem borer. On the day 2, the technical session was initiated, and Dr. N. Muthukumaran, Organizing Secretary, IQA 21, welcomed and introduced the lead speakers of technical session 3. The presentation on next generation fumigants for pest management in pre-planting and post-harvest situations was delivered by Dr. T. Swaminathan, Head, Research and Regulatory Affairs, Rasloka Services Limited, Australia. He elaborated the necessity of fumigation, mode of action and its types. Further, he apprised that the environmental deterioration was caused by methyl bromide, and he explained about the e-fume, ethyl formate, a product of Rasloka, which can be used as an organic fumigant. Dr. Binit Sharma, Assistant General Manager, Sulphur Mills Limited, has made his presentation on agrochemical registration and regulatory framework in India. He has given the key highlights of various protocol involved in the process of newer insecticide registration with Central Insecticide Board and Registration Committee, CIBRC, and types of registration and also the data requirements for registration. Dr. Claire Sampson, Technical Director, Russell IPM, United Kingdom, has presented the topic on management of Western flower thrips, Frankiniella occidentalis in strawberry. She explained briefly about the biology of flower thrips, also about the difficulties in controlling the pest. She emphasized that the use of blue sticky trap along with aggregation pheromone doubled the catch and reduced the population of the thrips in the field. And also the integration of the blue sticky trap will also helpful to natural enemies. Further, she enlightened the IPM approaches to manage the Western flower thrips. Dr. T. Nalini, Organizing Secretary of IQA 21, invited and introduced the lead speakers of technical session 4 to deliver the presentation. Dr. Naim Hassan, Managing Director, Russell Biosolutions Limited, United Kingdom, has delivered a lecture on practical use of insect pheromones for climate smart crop management. He highlighted on the use of biopesticides and pheromones in IPM, the growth in the use of various traps, namely fly catch traps, delta traps, and the need of incorporation in IPM module. Further, he stressed the importance of push-pull strategy in IPM. Dr. R.S. Suresh Kumar, National Manager, Product Development, Rossi Seeds Private Limited, India, has delivered a lecture on insights into insect resistance breeding. He narrated to exploit various defense mechanisms like antigenosis, antibiosis, tolerance, and escape in host plants against insects. And he mentioned that the presence of tuft trichophore and pilose type of trichomes were responsible for resistance in cotton. Further, he enlightened about the cutting edge technologies like transgenic technologies in resistance breeding of cotton. So at the end of each technical session, participants were actively involved in discussion with the lead speakers and cleared their doubts, shared their opinions, thoughts regarding the novel ideas in insect pest management. A total of 420 participants across the country were attended the workshop, out of which 360 participants from Annamalai University and 60 from other institutions across the country attended the workshop. The organizing team of IQA 21 hope that IQAC enabled two-day international workshop conducted during 7th and 8th September 2021 on the topic Changing Contours in IPM cater the needs of audience who attended this workshop offline and online. All the lectures given by the lead speakers were exhaustive, informative, and effective in imparting knowledge to audience in the current scenario and pest management. Thank you.
Thank you, Dr. Kadirvel. I now invite Dr. R. Kanakarajan, Director of this workshop, to propose formal vote of thanks.
evening, everyone, for giving me the memorable opportunity for proposing vote of thanks to all those who helped in conceptualizing and making it this event IQAR 21 into reality. I first convey my gratitude. First, I thank the university authorities for permitting us to take up the venture. I am elated and honored to thank my teacher, Dr. M. Kanabli sir, Dean, Faculty of Agriculture, for his wonderful inaugural address and his support for such initiatives. I wish to express my gratitude to our beloved head of the department, entomology of Dr. S. Sardimi sir, also Director IQC for his open-hearted motivation. I express my sincere thanks to my advisors and former head of the department and my teacher, Dr. B. Selman sir, Control of Examination, and my guide, Dr. S. Manikasam sir, for their guidance. For my fellow Director of IQAR 21, Dr. T. Selumuthukumaran, I express my thanks and congrats for his effort and energy in smooth conduct of the program. I wish to extend my regards and thanks to the convener of IQAR 21, Dr. C. Kadirvelu, for his outstanding support. I acknowledge with thanks the proactive role of all the organizers, secretaries, Dr. B. Anandaganesh Raja, Dr. M. Ramanan, Dr. N. Muthukumaran, and Dr. T. Nalini in the evergreen workshop of this institute, industry, interactive, and interface. On behalf of the team IQAR 21, I am happy to record my deep sense of gratitude to all the resource person for immediate acceptance and our motivation and invitation, making time to presentation in spite of the busy schedule, and for their wonderful and informative presentation. Thanks are due to Dr. Bhairappa, Dr. Ranganathan, Dr. Prabhu, Dr. Swaminathan from Australia, Dr. Benit, Dr. Claire Sampson, and Dr. Nayan from UK, and Dr. Suresh Kumar and Dr. Kannan, Dr. Luji from UK. So thanks for sirs for the wonderful presentations. I thank my colleagues in the Department of Entomology, Dr. R. Kannan, Dr. Ayasami, Dr. Satisilan, Dr. Chandasab, Dr. Palisami sir, for the unwavering support. In this connection, I would like to express my heartfelt gratitude to Dr. M. Prakash, Professor, Department of Genetic and Plant Breeding, Dr. Ramesh Kumar, Deputy Director, IQSC and Professor of Horticulture, Dr. C.T. Satappan, Associate Professor of Horticulture, Dr. Dhanasegar, Assistant Professor of Horticulture, for their support. The special thanks to Dr. Jayaprakash, Assistant Professor of Computer Science, for the super technical support in the conduct of this international workshop. I express my congratulations and regards to all the supporting staff of the department and faculty. Finally, I am thankful to all the esteemed delegates of the workshop from various parts of the Tamil Nadu and India for their participation and deliberations. I thank all my students, friends of Department of Entomology, PG and PG scholars for their active contribution to make this webinar grand success. Thank you, one and all.
So announcement for the participants. So the feedback form, once you are submitting it, we will be issuing you e-certificates for the students as well as the scholars. It will take 10 days time. I request all the participants to kindly bear with the time limit, and I request all of them to give the feedback. So the link is available in the chat box.